very much, Andrea, for that wonderful introduction. I want to say how delighted I am to be invited to speak to you here today. It's particularly exciting to me, at least, to be invited by postdocs and students. Not that it isn't exciting to be invited by faculty, but especially exciting to be invited by people at the beginning of their scientific careers. And so because of this, I thought that I would incorporate a little bit more of a personal narrative than I normally do in my talks. In other words, a personal narrative of along the lines of how did I get here to this stage, especially in my research interests. And I would also like to thank Andrea, who has been an absolutely fantastic host, and I've really enjoyed the giant diversity of people that I've talked to here. I'm really impressed by the breadth of, as well as the size of this department. Okay, so today I'm going to tell you a little bit about some of our research on the molecular evolution of visual pigment proteins, the protein in the eye that forms the first step in the visual transduction cascade that responds to light in all animals that can see. So this introductory slide here is just showing you the binding pocket where the 11 cis retinal chromophore sits. Okay, as Andrea alluded to, I've been interested in studying sensory systems and the visual system in particular ever since I was a young graduate student, what seems like oh so many years ago now. I was first inspired by a fantastic synthesis that I read in the Quarterly Review of Biology. I was very happy to see that that journal is actually still in existence, unlike a lot of other journals of its type. A fantastic synthetic review by somebody named Timothy Goldsmith at Yale that was entitled The Optimization, Constraint, and History in the Evolution of Eyes. So it's an incredibly broad synthetic review of the type that really doesn't seem to be written anymore. But this review really sparked a lifelong interest in sensory biology for me. And over the years, with my lab group, we have been able to investigate a wide variety of vertebrate systems, as Andrea alluded to. So in my lab, we're really interested in investigating some of the basic underlying molecular systems underlying vision, and especially the first critical steps in the visual pathway that underlies vision. We kind of see the natural world as a series, an unending series of mutagenesis experiments that we are taking advantage of in order to better understand the molecular mechanisms of vision. So we kind of look for model systems in nature that are likely to have produced interesting adaptive shifts in vision, and then we try and study those using a combination of computational as well as lab-based experiments. Okay, so some of the questions, some of the major sort of questions that we're interested in addressing are questions like how do sensory systems evolve and how the visual system has adapted to diverse light environments or visual needs of organisms. And as I alluded to, we're very interested in using comparative computational approaches to try and detect shifts in patterns of selection that might have occurred in response to shifts in ecology. And the thing that really ties this all together for us is that we're fundamentally interested in the mechanistic basis of sensory adaptations. So our main model system, as I've alluded to, is the visual pigment proteins and the proteins involved in the visual transduction cascade within the photoreceptors of the eye. So I'm going to talk about a couple of, I hopefully have time to talk about a couple of projects in my lab addressing these questions, including a project that we've had going on for a number of years now since I started my lab in Toronto on the evolution of snake photoreceptors, as well as I'm hoping to have time to talk about a collaboration with one of your newest members, who's unfortunately not here today, but of your EEB department, Hernan Lopez Fernandez, on the adaptive radiation of neotropical cichlid fishes. So first, let me start with our general approach to studying the evolution and adaptation of the visual system. As I alluded to, we like to study the molecular basis of potential adaptive evolutionary shifts by a blended sort of computational experimental methodologies. The key is basically using a class of computational likelihood methods called codon-based methods of evolution, models of evolution, to detect patterns of sequence variation that might be consistent with consistent with changes in selection. So really, this allows for us to capitalize on the availability of sequence data in the databases, as well as going out there and collecting sequences ourselves for interesting animals animals that use their vision in interesting ways. And it also, even more importantly, allows for the formulation and the testing of a priori hypotheses of selection, all in a phylogenetic framework. So this class of computational methods I'm talking about all arise out of likelihood methods of phylogenetic analysis and the use and implementation of codon-based models. This type of approach allows us not only to identify where in evolution changes in selection might have happened, right, in a phylogenetic context, it also allows us to pinpoint where in the protein might have been the targets of selection. And then what we can do is we can go into the laboratory, and so we can formulate very specific hypotheses about which amino acid substitutions might be associated with shifts in selection, and then go into the laboratory and use site-directed mutagenesis techniques to mutate those amino acid substitutions and see whether or not they result in functional shifts in the protein. So this is we think at least, a very powerful approach because it's basically a hypothesis testing framework in the context of evolutionary biology. So I'm talking a lot about these codon-based models of evolution. They have been in widespread use for a number of years now, and they basically involve this parameter here, which involves the rate of non-synonymous substitution over the synonymous substitution rate, or omega. And of course, this has been characterized, depending on values of omega, as being reflective of positive selection if the value of omega is above 1, neutral evolution if it's equal to 1, and purifying or negative selection if it's below 1. These models have, of course, been used, widely used, but mostly simply to detect the presence or absence of positive selection. In my lab, what we've done is we've tried to, go a step beyond that and to look at changes in evolutionary constraints. So not just, hypothesis testing to see whether or not there's positive selection in your data set, but also to look and see whether this DNDS ratio, omega, has changed over the course of evolution and how it has changed. And in order to do that, we've implemented a class of models called Clade models that were first proposed by Joe Bielowski and Ziheng Yang in 2004. And this class of models allow you to test for the presence of divergence selection. In other words, a class of sites for which this DNDS ratio has changed in one clade relative to another. This was first developed to detect differences in selective constraint as a result of gene duplications, but more recently, as my lab and other labs have shown, this is a very useful set of models for testing things. You can basically partition your data set any way you want, and instead of doing gene duplications, you could test, for example, aspects of ecology or physiology or organismal biology in this way. And then, as I mentioned, there's also the possibility of targeting amino acid residues at which it looks like the DNDS values might have changed, as reflected on this heat diagram here, and then use experimental methods to see whether or not these hypotheses that are generated using computational methods might actually result in changes in function in the laboratory. Okay, so that's our general approach. I'd like to just, because our model system is the visual system, I'd like to just give you a very brief introduction to the visual system, and then talk to you a little bit about some of the research that we've been doing. So as you may know, this is a diagram of the eye. Light enters through the cornea and the lens and hits a photosensitive cell layer at the back of the eye called the retina. The retina is a highly structured, layered organ of which, in vertebrates at least, the photoreceptor cells sit at the back. So light passes through this whole layered bunch of layers of cells and hits the photoreceptor cell layer, of which these highly specialized outer segments are full of the visual pigment. The visual pigment protein is the part of the visual system that's sensitive to light, that responds to light, and transduces the signal to the rest of the visual system that light has been perceived. We know a lot about rhodopsin structure because it was the first G-protein couple receptor to be crystallized. G-protein couple receptors are members of the immense pharmacologically important superfamily composed of thousands of members. This immense superfamily, the reason why there's so much interest in G-protein couple receptors is that over 30% of, I'm still amazed by this figure, over 30% of all drugs target G-protein couple receptors. And they're all thought to have this canonical structure where you have 7 transmembrane domains, so they cross the membrane 7 times, and they form this barrel-like structure, in this case, surrounding this red chromophore here, which is a vitamin A-derived 11-cis retinal chromophore. And that chromophore, which is covalently bound to the opsin, surrounding opsin protein, that chromophore is the only photosensitive part of this whole complex. It is the part that absorbs a photon of light, changes conformation, isomerizes in response to light, which then triggers a conformational change in the surrounding opsin protein. It basically muscles some of the helices out of the way and results in an opening. So as you can see, it pushes helices 5 and 6 out, and this results in an opening on the cytoplasmic face of the protein that allows for this heterotrimeric second messenger G protein to come and bind. And that results in the kicking out of the GDP, exchange of GTP for GDP, and that results in the activation of the signal transduction cascade within the photoreceptors of the eye that result in a signal that light has been perceived. So we also know something about the visual cycle. In other words, some of the conformational changes that happen after the activation of the rhodopsin protein. So basically, light activates the rhodopsin, and then in some of the fastest reactions known in the entire natural world, some of these steps take on the order of femtoseconds, right? It changes conformation that eventually results in the meta-2 biologically active form. And this meta-2 form is the form that activates the G protein transducin. After this is formed, with a certain time constant, it decays, the all-trans retinal is kicked out of the binding pocket, and it results in an empty pocket opsin form, which can then regenerate with 11-cis retinal. Now, the reason why I'm showing you is because we can measure the rates at which this active form is formed and the rates at which it decays, because they have different lambda max values, or they have different maximal absorbance values that can be measured using an absorption spectrophotometer. So in the dark state, before it's triggered by light, you can measure the absorption spectra of a visual pigment protein, and they all look something like this. These are just the 3 visual pigments that humans have for the red, green, and blue cone photoreceptors. So as you can see, they have this canonical absorption spectra that shifts into the UV when it's been activated by light. Okay, so vertebrates have evolved a duplex retina in order to cope with the orders of magnitude differences in light intensities that from the sunniest day to the darkest moonless nights, there's a difference in light intensities of more than 10 orders of magnitude. It's an incredibly difficult engineering problem to come up with a sensory system that can actually respond dynamically throughout that entire range, right? Usually at some point you will saturate and then you can't respond dynamically anymore. What the visual system has done in order to be able to respond dynamically is to evolve two highly specialized systems in one. One highly specialized system for nocturnal vision or low light levels, that would be the system mediated by the rods, and another that is for daytime vision. And so there are these trade-offs where with rod photoreceptors, you have things like increased photosensitivity, but at the expense of decreased temporal resolution. So you can basically perceive very small amounts of light, but you have to wait so long for the signal amplification to occur that you lose temporal resolution. Now, these different physiological properties are thought to be mediated by, at least in part, by the different versions of homologous gene paralogs that are found in rod photoreceptors versus cone photoreceptors. And the rod versus cone opsins are a great example of that, where rod opsins show some differences in biochemical function from cone opsins that seem to correlate with this physiological differences between rods and cones. Okay, so the two different projects that I want to tell you about today involve, first of all, rod versus cone photoreceptors in snakes. I want to tell you about the intriguing all-cone retina of garter snakes. And then I'd like to tell you a little bit, if I have time, a little bit about rhodopsin in neotropical cichlid fishes. So rod versus cone vision in garter snakes. It's been known for quite a long time that there are some snakes, including the garter snake, that have this really unusual all-cone retina. So only cone photoreceptors, no evidence of any rod photoreceptors. This is very unusual because it's thought that you can't really have good nighttime vision if you just give up all of your rod photoreceptors. And so for most animals, the vast majority of mammals and fishes and birds, they have both rods and cones in their retina. But in this case we decided to study the garter snake, which was an example of this. There was no evidence for any rod photoreceptors at all. And this was the work of a couple of graduate students of mine, Ryan Schott, who just finished and starting a postdoc at the American Museum, and Nihar Bhattacharya. So, not to give away the punchline, but we believe we have some evidence for the fact that there might be a transformed rod photoreceptor in the retina. So the thing is that if you look at snakes, it's been known for a while that some of the basal snakes actually do have rod photoreceptors, but then it looks like this may have been lost in some of the colubrid snakes, some of the Cynophidian snakes. So there could be two hypotheses for why we don't see any rod photoreceptors in some of these higher snakes. One could be because the rod photoreceptor is actually lost, right? And so you have this new cone class that could have been the result of a duplication of one of these other cone classes. Or it could be that second hypothesis that in fact the rod photoreceptor somehow was able to transform itself into a cone photoreceptor. I might say first off that there's very little evidence for this second sort of hypothesis, but we decided that we would take a look to see what actually, if we could determine which one of those evolutionary scenarios might be true. So the first thing that we did was to do microspectrophotometry, which is to look and see what wavelengths all of the different types of cone classes respond to, and this was in collaboration with Ellis Lowe. We found a UV absorbing photoreceptor, 360. We found a long wavelength photoreceptor, which is what we expected, and then we found this third cone class. We then looked in the, to clone all of the visual pigments, and we found that there were 3 different visual pigment genes, which included a UV absorbing, a long wavelength absorbing, but instead of another cone visual pigment, we actually found a rhodopsin. So that was our first indication that maybe this third cone class was actually a, could actually be a transformed rod. We did the scanning EM to confirm that indeed in this snake there is absolutely no evidence of a rod photoreceptor. All of the cone photoreceptors have very short, stumpy outer segments, so absolutely no evidence. So then, but we're still not believing that this could really be the case because there had never been an example of a rod opsin expressed in a cone photoreceptor before. So we did the, not only the in vivo or MSP on intact photoreceptors, we also took the gene and for the rhodopsin, and we expressed it in the laboratory by itself and measured its absorption spectra. And what we found is that the absorption spectra matched very nicely with the absorption spectra of the intact photoreceptors. So that was a pretty good indication that maybe, in fact, it was the same thing. There were some fine ultrastructural details that we were able to isolate through TEM that, in fact, one class of these cone photoreceptors, what looked like cone photoreceptors, in fact had very fine details of the ultrastructure that were kind of reminiscent of rods. And then we were actually able to, using an antibody, we were actually able to detect the presence of a rod transducin protein in the retina. So there's, we had a lot of circumstantial evidence that maybe these, this would be called circumstantial in a court of law. We didn't have the smoking gun yet, but we had some circumstantial evidence that actually, hey, maybe the rhodopsin is being expressed in rod photoreceptor cells, because we have the rod, another member of the rod phototransduction pathway that does not occur in cone phototransduction cascade, the cone G protein, in the snake retina. But we really wanted to have the smoking gun, so we decided, okay, we're going to do double labeling. We're going to use a rod opsin antibody and a rod transducin antibody, and we're going to see, are they actually in the same cells? And in fact, yes, we found the rod opsin expressed in the outer segment and the rod transducin expressed in the inner segments of the same cells. So we were able to show that in fact a class of cone photoreceptor cells in these snakes is expressing not just the rod opsin or rhodopsin protein. Those cells are also expressing another member of the rod machinery that you would normally find in rod photoreceptor cells, but they're both expressed in a cone, a subset of the cone cells. Now, why would these rods, what look like they have rod internal machinery, be masquerading as cones in these garter snakes? As I mentioned, it has been known that some of these basal snakes only have 2 classes of cones. They have the UV class of photoreceptor and the long wavelength class of photoreceptor. Now, if you notice, these 2 different classes of photoreceptors, they have their absorption spectra of almost no overlap. If you have almost no overlap, you don't have the capacity for real color vision, for true color vision. You just have 2 monochromatic input channels. And for these snakes that are highly visual, the addition of a third photoreceptor class is what gives them, it might give them the capacity for trichromatic color vision. So our best hypothesis at this point is that this might have been an alternative route to trichromacy. They basically co-opted the rods to function during the day to add a photoreceptor class where it was needed in order to fill out the spectrum and allow for trichromatic vision. So if this is true, if our hypothesis or if a model is true, we might expect, number one, we might expect to have adaptations in the rhodopsin protein to tune function to this new cellular environment that they're in, this cone photoreceptor environment, as well as number two, we might expect to see selection on other genes in the phototransduction cascade in order to work better with this, newly evolved sort of rod to cone transformation. So the first prediction, we actually looked, because that's one of the things my lab does, is look at rhodopsin function in the laboratory. We're actually able to show in another colubrid snake, the northern pine snake, that there were aspects of its function. As you can see in this snake, we also showed that there was a subset of cones that contained rhodopsin as well as rod transducin through the use of the same antibodies. But in addition, we were able to express it in the laboratory and measure aspects of its function that could tell us whether it was behaving in a more rod-like manner or a more cone-like manner. And that is through reactivity with something called hydroxylamine. And this is just showing that, in fact, more like conopsins, this rhodopsin was reacting with hydroxylamine instead of being resistant to it, which would be more like a rhodopsin. So that was the first hint that we were actually having a function of this rhodopsin that was actually more like a cone than would be typical of a rod. So what about, as I mentioned, what about the evolution of other phototransduction genes or other genes in the visual transduction pathway? Now this is a very complicated slide and I don't expect you to absorb all of it, but it's just to tell you that in fact the phototransduction cascade within the photoreceptor cells of the retina is something that is very well understood. We understand what all of the components are, we understand how they interact with each other, we understand how they're activated and how they're deactivated, how the process of light activation and how the recovery from light activation occurs. So we know what all of these components are and we can find them in the genomes of animals. So this is the work of my student Ryan. And what he did is he used, nowadays sequencing is now it's no problem to get huge amounts of sequence data. So he used a combination of data mining from draft genomes of reptiles, hybrid enrichment, in other words, sequence capture, using a set of curated genes from some of the snake genomes as well as from an Anolis genome to pull out the vision genes from taxa that have not been sequenced yet, as well as transcriptome data from whole eyes. So we took all of this and combined it with publicly available data to create a data set over representing 21 different species, including 13 species of snakes. And from those, each one of those species, we had a data set of 26 vision genes. So these are not just vision genes, they're genes involved in the visual transduction cascade of the photoreceptors that I showed you before, okay? Now, as you can see, one of the nice things about this data set is, for a lot of these genes, there are rod-specific components. That means they're only expressed in rods, as well as paralogous copies that are only expressed in cones. So that's divided into the blue and the red categories. as well as some genes that are expressed in both rods and cones, okay? And as I mentioned, we know exactly what all of these components of the visual transduction cascade do. So he isolated all of these, and then implementing the Clade models that I talked about before to examine the effect of transmutation on these genes, on the evolution of these genes. We used these Clade models to test for evidence for divergent selection between the cynophidian snakes, which have transmutation, or the evolutionary transformed rod to cone photoreceptors, versus all these other animals that don't show evidence for that. And what he found is that there was highly significant evidence for both positive selection as well as divergent selection in this group that has experienced the evolutionary transformation of photoreceptors. What was surprising to us is that the differences in DNDS or omega were in fact most pronounced in the genes in the cone phototransduction pathway. Those were more pronounced than the rod phototransduction pathway. So this was initially somewhat of a surprise to us, as we had been expecting to see selection on rod pathway genes, given all of what I told you before. However, this might be because we and others in recent years have been mostly focused on this aspect of the evolutionary transformation between an ancestral duplex retina with both rods and cones to an all-cone retina. Now, this is something that had been predicted over 60 years ago by someone named Gordon Walls, simply using a light microscope and looking at the details of the structure under a light microscope of photoreceptors. He, in fact, predicted that there was an evolutionary transformation of the rod into cones in snakes, but he also predicted that there is evolution of all rod retinas that happen multiple times, that are derived from all cone retinas. So it might be that we are detecting stronger selection on cone pathway genes because they may predominate due to these multiple evolutionary transformations toward nocturnality in colubrids and other snakes. Or the alternative is that it may be that the rod pathway genes are simply operating at their physiological limits. And I think that future studies of visual transduction gene function are going to be needed to address this. Okay, in the last few minutes, I'd just like to tell you about, I'd like to switch gears. I've been talking a lot about terrestrial systems, and I'd like to tell you a little bit about our work in aquatic environments. A lot of my lab in recent years has been working on vision in aquatic environments more and more. And this includes everything from cetaceans, whales, different types of whales, to high altitude fishes living in high altitude streams in the Andes. But the reason for this is because aquatic light environments show an extreme diversity that you really don't see in terrestrial environments. Now this beautiful figure is one that's put together by a joint graduate student that I have with Nathan Lovejoy, Alex Van Nijnatten. And here he is showing the differences in light habitats between marine habitats, clear marine habitats, the attenuation of light as you go down with depth compared to some of the major rivers in South America. Now, Alex's interests are in marine-derived transitions from marine to freshwater systems, and so that's why he put this together. But some of the striking things about these differences are that you can see here this is 800 meters, whereas this is only 2 meters. So light penetrates in ocean environments much deeper than it does in freshwater environments, typically. And the diversity of freshwater environments is extreme. Now, it's known not just that light tends to penetrate much further in oceanic environments, but it also becomes blue shifted. It also tends to get blue shifted with depth. The reverse is true with most freshwater environments, where as you go down, the light spectrum, the spectrum of available light tends to get red shifted. So light is attenuated much more in rivers, and it tends to be red shifted and much dimmer. So I'm going to spend the rest of the time on my talk on the work of Frances Hauser, who was a joint graduate student with myself and Hernan, who just moved here. Her interest was on investigating neotropical cichlids and vision in neotropical cichlids. Neotropical cichlids, although you might know lots about them now that Hernan is here, they're perhaps not as well known as their relatives, the African Rift Lake cichlids, but they're also thought to be an adaptive radiation in terms of morphology and feeding strategies and body shape, and they are particularly diverse, as you can see here, in terms of coloration. Frances was interested to see whether this radiation in geotropical cichlids resulted in shifts in selection in any of the vision genes. She isolated visual pigment sequences as part of a larger phylogenomic study that was happening in Hernan's lab, where he was doing a large next generation sequence capture study in which 2,000 exons were captured for phylogenomic purposes. And among those 2,000 exons were visual pigment genes. So this is just a side note to say that because of our involvement in this collaboration, we were able to obtain a really nice sampling across this whole group of cichlids, because it was also part of this genomic study. And we were also able to have, because there were random samples from the genome, control genes to which we could compare our results to make sure that what we were seeing in the visual pigment genes was actually, characteristic of visual pigment genes and not just throughout the entire genome. All right, so we had, I'm going to just report, she's done all of the visual pigment genes now, but I'm just reporting rhodopsin right now. She had excellent sampling of rhodopsin across 101 species of neotropical cichlids that span the entire diversity of the clade. So we could use this to test different hypotheses of evolutionary divergence in visual genes using these clade models that I told you about. And we tested a bunch of different clade models, some that are simply based on the fact that different cichlid tribes have very distinct morphologies and traits and feeding ecologies, so we separated them out like that. Previous studies from our labs and others had shown that there are differences between lake and river cichlids in terms of their evolutionary rates. And then finally, we tested one which involved biogeography and the invasion of Central America by these cichlids as opposed to South America. And what we found when we tested all of these different models is that the best fitting model, as determined by AIC, was found to be the model that partitioned the tree between Central and South America. So this model showed extremely high rates of positive selection in the Central American partition. Omega was above 14 in that partition, as well as, so high rates of positive selection, right? As well as high rates of divergent selection between Central America and South America. So that part is also statistically significant. The control genes that we looked at from other genes in Hernan's data set, Hernan and Katrina's data set, did not show any evidence for either positive selection or divergent selection. And when we look at the distribution of omega values across the gene, across different sites in the protein, what we saw is that there were some sites that came up as positively selected only in the Central American partition. So as you can see, some of these sites here. And so what we did is we took some of those sites, like 83, and we went and we tested them in the laboratory. So I just want to talk a little bit more about 83. 83 was identified not only as a target of strong positive selection, only in the Central American partition, not South America. It was also found when we traced these amino acid substitutions onto our cichlid phylogeny. It was found that this site, 83, transitioned from asparagine to an aspartic acid multiple times. So it was found to undergo independent transitions from asparagine to aspartic acid. Now, aspartic acid is a charged residue, and asparagine is not. Charged residues are really unusual in membrane proteins, and this one occurs in a key region of the rhodopsin protein that's within the chromophore binding pocket. That part of the structure is tightly packed. It's proximal to the chromophore, and previous studies from our lab and others had suggested that this residue might affect rhodopsin function. So we decided to test the effect of this particular amino acid substitution by expressing it in vitro. Now, just to very briefly give you an idea of how we do this, we take the gene and we transfect it. We put it into this expression vector. We transfect it into cells. Those cells make lots of the protein for us. We purify it, and then we subject it to a number of spectroscopic assays. One of those assays, in addition to determining lambda max, is to look at light-activated retinal release. How long does the light-activated state last? Okay? And what we found is that there were significant differences in this phenotype when we looked at the mutant versus the wild-type version of this protein. So we took the wild-type version that has asparagine, the uncharged residue, we mutated it to the charged residue, and we saw a significant shift in function toward faster kinetics, so faster retinal release. I'm going a little bit faster because I'm running out of time, but we compared this to the control genes that we had. Bovine rhodopsin is the standard control for these in vitro assays of rhodopsin because it is best understood. We also, as a control, use zebrafish rhodopsin, which my lab has done a lot of work on as well. So we could show that basically there was a shift in function when you introduce this positively charged residue that resulted in faster kinetics. So our computational analyses, basically they indicated markedly higher evolutionary rates in the Central American cichlids, which suggested an acceleration of divergence upon colonizing these environments. Our experimental studies suggested that this is associated with an increased rate of retinal release or faster rhodopsin kinetics. These faster kinetics suggest an adaptation to the increased light levels that occur in the clear waters of Central America. So we do know that Central America, unlike South America, tends to be dominated by these small clear streams, whereas in South America there's a much bigger diversity of light environments, including some of the major rivers that actually, where light attenuation is much stronger. So in the future, we're looking at other vision genes in these fishes to see if they show similar or different patterns. So I hope that I've been able to convince you that we can use diversity in natural systems to better understand some of the molecular mechanisms underlying visual adaptations in response to shifts in ecology and organismal phylogeny, and that computational methods, codon-based models, such as those implemented in some of the Clade models and other lineage-specific models, can be used to detect interesting changes in the patterns of selective constraint in evolution, which can then be taken into the laboratory and tested. I'd like to just acknowledge some of the people in my laboratory that were involved in this work, including a lot of these people were involved in the snake work that I talked about, including Clement Hermina, Mungshu, Natalie, Ryan, and Nihar. And then Francis was involved in the cichlid project along with Hernan, and Vince Tropepe has been one of our long-term collaborators with the immunofluorescence studies. Thanks for your time. I'd be happy to take any questions. Yes?
You have considered a range of attenuated light levels in these fishes. And I just wondered if you would find fishes that were taxonomically close to the ones that you have studied, but have become blind in caves. If you were to look at their genomes and find out what kinds of differences there were and what those differences there were in the phylogeny.
Yes. Yeah, in fact, cave fishes are really interesting, and there's a lot of people who are interested in studying the mechanisms of how they go blind and exactly what happens to the proteins in the eye. One of the surprising things in many of those studies is that, in fact, the visual pathways are largely intact, and they're largely similar to species that are not blind, or even fairly closely related populations of fishes that are not blind. And in many cases, what they found is that, in fact, there are often populations within the same species, and some of which are cave-dwelling populations and other which are surface-dwelling populations. And in fact, there's some gene flow between the two. So, yeah, so it seems like it's much more complex than people at first appreciated. Yes. Yes, yes, this is true. I mean, snakes are incredibly diverse, and you know, so some of them tend to be more visual than others. Some of them have larger eyes than others. So I think that it is you know, at this point, we're really just at the tip of the iceberg of what we can explore in snakes. We sort of just took the, you know, took the evolutionary question that sort of most stumped us and focused on that one first. But snake vision is definitely one of the most interesting, and then the evolution of sensory systems in snakes is one of the most interesting questions out there. So, yeah. All right. Thank you. Yeah. One more question?
Is the trade-off ever in favor of having better day vision, giving up on night vision, instead of something like a hawk? They definitely use good day vision. Do they have poor night vision as a result?
Yes. That is a very interesting question. There are some examples, even within mammals, like, for example, diurnal. Some diurnal squirrels have only cones in their retina. And yeah, it does seem like there are rare instances where the trade-off does work in that direction, and it probably has something to do with high acuity vision during the day. But it is exceedingly rare because, in fact, you don't even need that many rods, right? Rods are not about having high acuity vision for most animals. You can still, you know, have enough photoreceptors if you have a very sparse sampling of rod photoreceptors. That's enough to drive the ability to see shadows at night. And so that's why it seems like most animals just, you know, it's better not to give up your rods. But absolutely, you know, it's nature, so there's always an exception anywhere if you look hard enough. Okay, thanks for coming.